Hello, my name is Joshua Mugabi. I welcome you to topic 11.1, Inheritance. We're going to look at learning outcome A. Understand the process of cell division and the significance of meiosis. We are starting with learning activity. In groups or pairs, learners view any animation on meiotic cell division. Research using other sources and then identify the stages and describe what occurs at each phase using annotated diagrams and notes. Item 1, meiotic cell division. Let's start with microscopic observation. Students observe a prepared slides of germ cells from a flower anther under a microscope. Different cells are seen at various stages of division. We have the first cell here, the second cell, the third cell, and the fourth cell. Tasks. Identify the stage of meiosis shown in each cell. Identify the stage of meiosis shown in each cell. We are starting with prophase. Our cell here. It is saying 2N. 2N means it is a diploid and it is being represented by 2 pairs of homologous chromosomes. We have this pair and we have this pair. But there are supposed to be a total of 23 pairs of homologous chromosomes. But because the space is very small, the 23 are represented by 2 pairs of homologous chromosomes. And it is a diploid, 2N, prophase 1. Then we have another stage which we are calling metaphase. It is this stage here. And in metaphase, we are seeing the homologous chromosomes aligning on the spindles. That is metaphase 1. Homologous chromosomes align themselves on the spindle. We have another one, and this is anaphase 1, this stage here. This is cell. The homologous chromosomes are separating and they are moving towards opposite poles. Then we have the telophase 1. Telophase 1 is this stage here. Here, the homologous chromosomes have separated and they have gone to different poles, and the cell is starting to divide. This one is referred now to as haploid. Number 2, describe the events occurring at each phase. Describe the events occurring at each phase. We are starting with the phase we called prophase, this phase here. And what is happening? The homologous chromosomes pair up, what we call synapses. Now, when you look at it here, you are going to see the homologous chromosome, these two, pairing up, and also you will see this second pair also coming and pairing up. These ones are what we are calling the homologous chromosome. One came from the mother, another one came from the father. Mother, father, but they are pairing up. There are 2 pairs. They're supposed to be 23 pairs. But because of the space, we have represented the 23 by only 2 pairs. So this is what we are calling 2N. It is a diploid. They are making up of a total of 46. Those are 23 pairs. But here we are having 2 pairs. So these are the ones we are referring to as homologous chromosomes, and they are pairing up. That process of pairing up is what we call synapses. Then, at the same time, crossing over occurs between non-sister chromatids. Now, I want us to look at this one very carefully. If you look at these ones, what we are calling the homologous chromosome. Let us first look at this homologous chromosome here. This homologous chromosome, we have this one is a homologous, is a chromosome, and this one is a chromosome. But each chromosome has got a sister. They are what they are calling sister chromatids. So if you look at this one has got this one here, that is a sister chromatid, and it has got also another sister here, this one. Those 2, we call them sister chromatid. This one and this one, it is the same chromosome made up of sister chromatids. And this one and this one, they are chromatid of the same chromosome. So what we are saying here, crossing over occurs between the non-sister chromatid. So when you look here, you will see that this chromatid here of this chromosome and this chromatid here of this chromosome, they are crossing over. And that place where they are crossing each other, let me make a circle here where they are crossing. You can see that they are crossing here. That crossing point there is what we are calling crossing over. And these ones that are crossing over, they are called non-sister chromatid because they are coming from different chromosomes. So they cross over each other. So during prophase 1, that is when we have crossing over taking place between non-sister chromatid. Non-sister chromatids, these are chromatids coming from different chromosomes. Then still under prophase 1, there is what we are referring to as the chiasmata. Chiasmata become visible. Now, the chiasmata is that point where crossing over takes place. When the two cross over like that, that point of crossing over is what we call a chiasmata. Let's take this one to be a sister chromatid. If that is a sister chromatid, and it is crossing over with another non-sister chromatid. These are two. There is this one. Those are called the red ones. This one and this one, they are called sister chromatids. Then we have another one here. Now, this one and this one, they are sister chromatids. This one and this one, they are sister chromatids. The two chromosomes, they are homologous chromosomes. Now, this is a sister to this, and this is a sister to this. But this one is crossing with this one, who are not sister chromatid. So those ones, we called them non-sister chromatids. And when they're crossing over to each other, that is what we are calling a crossing over here and here. That is a crossing over. And now that crossing over point here, this crossing over point is what we refer to as the chiasmata. Then another point, the nuclear membrane breaks down and the spindles fibers form. So when you look here, you will see that this nuclear membrane, which was surrounding the nucleus, it disappears, and then also the spindles start forming. So we form the spindles, but the nuclear membrane starts disappearing. Another one is metaphase. The metaphase. Now, our metaphase, what happens is that the paired homologous chromosomes, which we call the bivalent, align at the equator. So when you look here on our diagram here, you will see that the homologous chromosomes, these homologous chromosomes, and also these other homologous chromosomes here, they align themselves on the same spindle. If you look at these homologous chromosomes, they align themselves on this spindle here, and this homologous chromosome here align itself on this spindle. So that's what we are saying, that the paired homologous chromosomes, the bivalent, align at the spindle. Then the spindle fibers attach to the centromere. The spindle fibers attach to the centromere. Now the centromere is this middle part, the middle part of the chromosome. That is what we call the centromere. So the spindles are going to attach themselves on the middle part of the chromosome, which we call the centromere. So the spindles will attach on the centromere, and that one takes place at the metaphase stage, metaphase 1. Then we have another stage, which is anaphase 1. What happens in anaphase 1? The homologous chromosomes, they separate and move to opposite poles. So let us look at anaphase 1 here. When you look at anaphase 1 here, you will see that these two homologous chromosomes, they are separating and moving to different poles. This one goes this side, and this one goes the other side on another pole. The same applies to this homologous chromosome. This one moves this way, and then this one moves that side. So that's what we are saying, that the homologous chromosomes separate and move to opposite pole. And this one is taking place in anaphase 1. We started in metaphase 1 when they are aligning at the spindle here in the equator here. Now they are moving to opposite pole. That is in anaphase 1. Then the centromere does not split. The centromere does not split. Where is the centromere? The centromere is on a chromosome. And a chromosome has got what we call sister chromatid. And those sister chromatid are attached at a point we call the centromere. And where is that point? In the middle there. That is the centromere. So the centromere of the chromosome, does not split during anaphase 1. So that's what we are saying here. Centromere do not split in anaphase 1. Then we go to telophase. In telophase, this is telophase 1. Here in telophase 1, the chromosomes reach the pole. So here we can see in telophase 1, the chromosomes have already reached the different poles. They have separated and they have reached at the different poles. And then also the chromosomes, the nuclear membrane may reform. So we can see here that the nuclear membrane has restarted forming. They are forming a nuclear membrane around the chromosomes. And then lastly, cytokinesis occurs, forming two haploid cells. So what we are referring to as cytokinesis is whereby the cell now is going to divide into two in the middle. That part here, that is what we are referring to as cytokinesis. The cell is going to divide into two, and we shall form two haploid cells. Item 2, meiotic cell division. We are going to emphasize on cell division. A diagram shows chromosomes arranged singly at the equator of the cell, with spindle fibers attached to the centromere. So we are having here our cell. So we are having this cell here, and when you look at this cell, you will see that the chromosomes here, each chromosome is attaching itself on its own spindle. Look at this chromosome, it is attached to this spindle. This chromosome is attached to this spindle, and this one is attached to this spindle. And in the middle of the cell, that is what we call the equator. This one is referred to as the equator. So here we are saying that the chromosomes are arranged singly, meaning one at a time, at the equator. So our equator is this point here in the middle. The point in the middle here is what we call the equator. Point where the chromosomes are aligning themselves is the equator of the cell, with the spindle fiber attached to the centromere. So the spindle fiber, there are these fibers moving from this pole to this pole. Those are the spindles. And the spindle fiber are attaching at the centromere. The centromere is the one in the center of the chromosome, or which attaches both sister chromatids together. So that's where the spindles attach themselves. Task: identify the stages of meiosis shown. They want us to identify the stage of meiosis shown. So which stage is this one shown here, where the chromosomes align themselves singly at the equator and each one is attached on its own spindle? That stage is referred to as metaphase 2. Metaphase 2. State 2 events occurring in this stage. State 2 events occurring in this stage. Which 2 events are you seeing occurring in this stage? 1. The chromosomes align up singly at the equator. So one of the events that we are seeing, that each chromosome, which is made up of 2 sister chromatids, align itself at the equator and it is on a single spindle. All of them are aligning themselves on the equator. And I told you that the equator is the middle of the cell where the chromosomes are arranging themselves. So you can see that the chromosomes are aligning themselves at the equator and each one is alone. They are not homologous, they are not paired chromosomes, but each one is aligning itself alone at the equator. Then the second one, the spindle fibers attach to the centromere. So you can see that the spindle fibers, they are going to attach on the centromere in the middle here. That's where the spindles are going to attach themselves on each chromosome. Where you have the centromere, a spindle fiber will attach. So that's what we are saying. The spindle fibers are going to attach themselves on the centromere. So those are the 2 events that are taking place. 1, the chromosomes are aligning singly at the equator, as you are seeing them here at the equator. And then secondly, the spindle fibers attach to the centromere. So every chromosome, a spindle fiber will attach at the centromere. Item 3, still on meiotic cell division. We are going to compare 2 stages. We want to compare 2 stages. 2 cells are shown. Cell A shows chromosomes separating, but centromeres remain attached. This is one here, cell A. And cell B shows sister chromatids separating. In cell B, we are seeing the sister chromatid separating. I want us to analyze it very clearly. Cell A, we are seeing the chromosomes. There were two here, and one is going to one pore and another one is going to one pore. And in cell B, we are seeing a cell here. It had a chromosome in the middle, and one chromatid is going the other side and another chromatid is going the other side. Now let us go to our task. Task: identify the stages represented by cell A and cell B. They want us to identify the stages represented by cell A and cell B. What are those stages representing cell A? The cell A is this one here, and which stage is representing cell B, this cell here. So we can identify them. So cell A is being represented by anaphase 1. And what is happening in cell A here? We said that in cell A, we are seeing that the chromosomes are separating, but the centromeres, they remain intact. So in cell A, the chromosomes are going to different poles. And when they are going to different poles, it is the chromosomes separating, but their centromeres remain attached. Then in cell B, this one represents anaphase 2. Anaphase 2. Our cell B here represents anaphase 2. And what did we say in anaphase 2? The cell here, the sister chromatids are the ones separating. Sister chromatids are the ones separating. So when you see here, in the middle it was one chromosome. But now it's split into two parts. When it splits into two parts, one chromatid goes the other side and another chromatid goes the other side. So that's why we are saying that the sister chromatids are separating. And the sister chromatid to separate, that means that we are in anaphase 2. Describe one event in each stage. They want us to describe one event in each stage. Can we describe one event that is taking place in this one? We have called it anaphase 1, and one event that is taking place in anaphase 2. Can you look at those events and describe them? So in cell A, which is anaphase 1, here we are seeing that it is the homologous chromosomes which are separating. And when they separate, their centromeres do not divide, as we have explained earlier. Two homologous chromosomes are separating, going to one pole and another one going to another pole, but their centromeres are not dividing. That is in anaphase 1. Now, when you look at cell B, that is anaphase 2. Here, cell B, this is anaphase 2. Here we say that the centromeres split and the sister chromatids move to opposite pole. So here what we are seeing, it was one chromosome in the middle here, which had two sister chromatids. Now, when they started separating, the centromere in the middle split. So we are saying the centromere split, and when the centromere split, the sister chromatids are separated. And when the sister chromatids are separated, they move to opposite pole. That is the event which we are seeing in anaphase 2. Item 3. We are still in meiotic cell division. Completion of a table. We want to complete a table. Students are given an incomplete table on meiosis. So we have our table here. It has the stages and a description. We have prophase 2. Prophase 2, there is no description what is taking in prophase 2. So we have to complete this part. And then we have these two spaces here. We don't know which phases are these or stages, but here they are saying a chromosome aligns at the equator singular. So we can give this phase here. And here there is a sister chromatid separate and move to opposite pole. Which stage is this one? And the last stage they are saying is telophase 2, but they have not given us the description. We have to fill this part here. Okay, so our task. Let's look at our task. Our task says complete the table by naming the stages and describing what occurs, the event. So there we are. We are starting with this stage here. We have to fill this part. What can we fill there? That is, they are saying it is prophase 2. Which description? What happens in prophase 2? So in prophase 2, what is happening is that the chromosomes condense. By condensing, we mean they reduce in number, as you are seeing right here. You can see that the chromosomes become visible. And then also the nuclear membrane breaks away or breaks down. So during that time of prophase 2, this nuclear membrane which is surrounding the chromosomes is going to break away. And after that one, then the spindles fibers are going to form. So the spindle fibers are these ones on top. They will start forming here, the spindle fiber. So in prophase 2, what happens is that the chromosomes condense, which were thread-like structures. They become shorter and can be easily seen as chromosomes. Then the nuclear membrane breaks down. So this nuclear membrane is going to break down during prophase 2. And then the spindle fibers will start forming. Spindle fibers are these ones that we are seeing from one pore to another pore. They are starting to form. That is the description of the events that take place in prophase 2. Then we go to our stage, another stage. And this stage, remember they gave us this space here. The space, we don't know the stage. But here they are saying that in this stage, what happens is that the chromosomes align at the equator singular. As we are seeing in the diagram here, you can see that each chromosome is aligning alone at the equator, and it is attached on the spindle alone singly. But all of them, they are aligning along the equator. So which stage is this one? Which stage is this one? Where the chromosomes align at the equator singly. That stage is metaphase 2. So in metaphase 2, that is when the chromosomes align at the equator singular, like the way we are seeing it here. Each chromosome is aligning itself at the equator alone, not in pair. When that one happens, we know that that stage is metaphase 2. Now we have come to another stage. This stage, we don't know which stage, but they are saying that during this stage, the sister chromatids, they separate and they move to opposite pole. We don't know the stage, but when you come this side, here you are going to see what they are saying here. The sister chromatids are separating. Here you are seeing the sister chromatids have separated. This one and this one were sister chromatid. This one and this one were sister chromatid. They have separated. And what do? They move to opposite pole. They move. This one moves this way and this one moves this way. These ones were on one chromosome. Now they are separating. This one is moving this way and this one is separating this way. Now, which stage is that one where sister chromatids separate and move to opposite pole? Which stage is that one? That stage is what we call anaphase 2. So this stage what we are seeing here is anaphase 2. And what happens during anaphase 2? The sister chromatid, they separate and they move to opposite pole, as we are seeing them here, separating and moving to opposite pole or in different directions. That is anaphase 2. Then we go to another stage they are calling telophase 2. Telophase 2. What happens during telophase 2? We don't have any description here of events that are taking place in telophase 2. What is taking place in telophase 2? Can you try to describe the events? In telophase 2, the chromatids reach the poles. So we are seeing here in telophase 2, when you look here, the chromatids have now reached the different poles. And when they reach the different pole, the nuclei reform. So we can see that here the nucleus are starting to form. This nucleus will form around the chromosomes, and this nucleus will form around the chromosome. And now we have 4 haploid cells have been formed. So because this one was a single cell, we have got 2 cells coming out of it. But remember, they were 2 single cells, and when division takes place, they become 4 haploid cells. So that is what takes place during telophase 2. Item 4, meiotic cell division. We are still emphasizing on genetic variation. A teacher explains that meiosis increases genetic variation among offsprings. Meiosis increases genetic variation among offsprings. Now here, before we continue, there is this variation. Variation is the change or the difference appearance in organisms of the same species. For example, when you look at these 2 elephants, it is the same species, but you can see that they are different from each other. That is what we are calling variation. So here the teacher explains that meiosis brings genetic variation among offsprings. So let's go to our task. Identify the stages of meiosis responsible for genetic variation. They want us to identify those stages of meiosis that are responsible for genetic variation. Why are these 2 elephants different? And if the teachers explained that it is meiosis which increases genetic variation among offsprings, so which stages of meiosis is responsible or are responsible for this genetic variation? So the stages involved is prophase 1 and metaphase 1. So this variation that we are seeing in the elephants being different, yet they are of the same species, that process is brought about by meiosis, or this variation is brought about by a process of meiosis and during the stages of prophase 1 and metaphase 1. Describe what occurs at those stages. They want us to describe what occurs at those stages. Remember, we have said that the stages of meiosis which bring about variation, we have meiosis, prophase 1 and metaphase 1. So they want us to tell us what takes place in prophase 1 and what takes place in metaphase 2. So they want us to describe what takes place there. We start with prophase 1. In prophase 1, there is what we call crossing over happening between two homologous chromosomes. So what happens in prophase 1? Remember, we said that in prophase 1, what takes place is that homologous chromosomes align themselves together. Homologous chromosome, what we call homologous chromosome, one chromosome comes from the paternal and another chromosome comes from the maternal, but they are responsible for the same characteristics in that organism. So they come and align themselves like what we are seeing here. Okay, we have this black one. Let us say this one is from maternal, and then we have this white chromosome coming from the paternal. Now these two chromosomes, when they come together like this, they align. Remember, we said that each chromosome, like this black one, has got two chromatids attached at the centromere. Those chromatids, this one and this one this way, we call them sister chromatids. So these are sister chromatids from the maternal, and these are sister chromatids from the paternal. Now when they align themselves, the non-sister chromatid, this chromatid here from the father, comes and crosses with another chromatid from the mother. And at that point where they cross, this point here, that is what we call crossing over. Crossing over occurs between homologous chromosomes. These two chromosomes, this one and this one, they are called homologous chromosomes. But they have got what we call chromatid. So this chromatid of this chromosome comes and crosses with this chromatid of this chromosome. And at the point where they cross here is what we call the chiasmata. So that happens in prophase 1. In prophase 1, crossing over occurs between the homologous chromosomes. Then in metaphase 1, in metaphase 1, what happens is that there is a random assortment. There is random assortment of homologous chromosomes at the equator. random assortment of the homologous chromosomes at the equator. Now I want to explain this one a little bit slowly so that you can understand. Now we are in metaphase 1. And if you recall very well, we said that in metaphase 1, what happens is that the chromosomes align themselves at the center of the cell. And that center of the cell, we call it equator. They align themselves at the equator. So remember, in metaphase 1, we have homologous chromosome. We said that there are two chromosomes. One came from the mother, and another one came from the father. So when they come to align themselves here, this one came from the mother, and this white one came from the father. And this one came from the father, the white one, and this one came from the mother. So the homologous chromosomes are aligning themselves on the equator. Now I want you to see what we are calling random assortment. We have divided the cell into two along the equator. Now we can say, if we say the upper part, we can call it part A. Okay, let's call the upper part as part A, and we call the lower part as part B. Now I want you to see how these chromosomes are arranging themselves. Look at these ones. You see, this one came from the mother. It has aligned itself towards part B of the cell. And the one from the father has aligned itself towards part A of the cell. Now look at the second pair. The second pair, when it aligns itself, the one that came from the mother has aligned itself towards part A of the cell. And the one that came from the father has aligned itself towards part B. So that is what we call random assortment. Random assortment means random arrangement. When they are arranging themselves here, there is no rule that says that those who are coming from the mother, the black ones, align yourself on one side, like side B. And those ones that are coming from the father, align yourself on the other side of the cell part A. So you can see that they have arranged themselves independently. And this is metaphase what? Metaphase 1. So when they start separating, this one, the one which came from the mother, will go this side. See? It will go this side. And the other one that came from the father will go that side. And when you look at the second pair, the one that came from the mother will go that side. And this one that came from the father will go this side. So you can see that on this side we shall have one from the mother and one from the father. And also the other side, one from the mother and one from the father. Meaning that there wasn't any arrangement, that those ones that are coming from the mother arrange this side. Those from the father arrange the other side. So that is what we call random assortment of homologous chromosomes at the equator. They just arrange themselves anyhow, no order. That for us who came from this way, we come this side, and for us who came this way, we come this side, because at the end, we are going to go the other side. So because of that arrangement, that is the one which is going to bring about variation among the offspring, because they are different arrangement of homologous chromosomes. They assort themselves independently, or what we refer to as random assortment. Now let us look at the summary. Now, in our summary, we can say that meiosis consists of two divisions, And the first division is referred to as meiosis 1, and the other one is meiosis 2. Then we have also seen that in meiosis 1, this one separates the homologous chromosome. In meiosis 1, it is the homologous chromosomes separating themselves. And in meiosis 2, it is the sister chromatids that are being separated. And this one results into a final result of 4 genetically different haploid cells. So the cells that we are going to end up forming, they are going to be genetically different. And that is what brings about variation. Now, our focus here is mainly to identify the stages of meiosis. We have been trying to identify the stages of meiosis, and we have said that the stages of meiosis, we have got different stages which we have gone through. And then also we are mainly focusing on the description of the events that are taking place in each phase. I think by this time you can easily now recall the stages of meiosis and the events that take place in those phases. Thank you very much for participating in topic 11.1. This is inheritance, and we have been looking at learning outcome A, understand the process of cell division and the significance of meiosis. That is our learning outcome. So what have we been specifically looking at is what we are referring to as our learning activity. Here we are saying in groups or pairs, learners view an animation or meiotic cell division. They do research using other sources and then identify the stages and describe what occurs at each phase using annotated diagrams and notes. So here we have been mainly emphasizing on identifying the stages and to describe what occurs in each phase, which I hope all of us now, we are very conversant. Please visit me at Mugabi Joshua Biology on YouTube. Don't forget to like, to share, and subscribe.